All right. Good morning, everyone. Welcome to our workshop session, Model System to Study Non-Pulmonary Manifestation of CF. My name is Kyushik Moon. I'm an assistant professor at Cedars-Sinai Medical Center in Los Angeles. And my other chair over here is Benjamin Rose, who is an assistant professor of microbiology, immunology, and orthopedics at the Dartmouth Geisel School of Medicine. Please be aware of this disclaimer that the information provided during this conference is intended to be educational and informational. This session will focus on cutting-edge model system designed to study non-pulmonary manifestations to better understand the effect of CFTR on different organ systems, as well as to provide better context on various aspects such as infection dynamics, microbiological interactions, immune responses, and understanding CFTR biology for the path to a cure. These are our objectives. With that, we have a great panel of speakers here today. Please accept my apologies if I mispronounce your name. Our first speaker, Dr. Agnieszka Swiatecka-Urban, who is a physician scientist and a professor of pediatrics at the University of Virginia School of Medicine. Yelling Yi is a research manager in Dr. John Engelhardt Group at the University of Iowa. Unfortunately, she couldn't attend this conference this time, so Dr. Engelhardt will be giving the talk on her behalf. Dr. Engelhardt is a professor of anatomy and cell biology and has made tremendous contributions to the CF field by developing CF pheromone models. And Dr. Patricia Scott is an assistant professor of biomedical science at the University of Minnesota. Ted Neff is a PhD student working with Dr. Ling Yang, who is an associate professor of anatomy and cell biology at the University of Iowa. And our last speaker, Oscar Omar Morales, is also a PhD student at the University of Saskatchewan in Canada. He works with Dr. Veronica Campanucci, who is an associate professor of anatomy, physiology, and pharmacology. Congratulations. Our session will be structured as a 15-minute presentation followed by 8 minutes Q&A. And audience can ask questions either ways by using loud voice and the mobile app NACFC 2024. Okay, so please welcome our first speaker, Dr. Agnieszka Swiatecka-Urban.
Thank you. First, thank you to the organizers and the chairs for invitation to present. And I have nothing to disclose. So the novel therapy for CF lung disease not only improved longevity, but also uncovered the extrapulmonary manifestations, their high prevalence and severity, shifting the care from preventing mortality to preventing morbidity. And one of the emerging challenges for the aging population with CF is chronic kidney disease. Each of us is born with certain nephron endowment, so we have to use them wisely because we don't make any more nephrons after we are born. And each of our kidney contains between 0.5 and 1 million of glomeruli, which connect with the renal tubules, where the function between the different nephron segments and the glomeruli perform very important functions in the electrolyte and acid-base balance, organic compounds, and nutrient transport, as well as excretion of waste products. Kidney function is measured by the estimated glomerular filtration, which in a healthy young person ranges between 90 and 125. And there is an expected age-dependent decline in kidney function. And studies from the general population demonstrated that the median yearly decrease in eGFR is approximately 1 ml per minute per 1.73 meters square after we reach age 30. So what it means for all of us is that by the age of 60, we're expected, if we start with a GFR of 125, the maximum normal, that our GFR is going to decline to the level where it will be borderline with CKD, so anything less than 90. And it continues. This relationship may shift leftwards if we start with GFR that is less on the spectrum instead of 125, but lower. So symptoms of chronic kidney disease develop slowly and silently until the disease is very advanced. And most people who have mild forms of chronic kidney disease don't even realize that they have the condition. So moderate CKD, or stage 3, we stage them between 1 and 5, corresponds to the GFR of 30 to 60, and it is experienced by many people in the general population without even knowing. In people with CF, a study by Quenan from 2011 demonstrated that at that time, the age-adjusted prevalence estimates for CKD3 were twofold higher than in the general US population. In addition, the annual percentage of prevalence of CKD3 was 2%, and it was doubling every 10 years of age increase, and it reached 19% in people over 55. What it means that in 2011, or by 2011, 1 in 5 people with CF age over 55 had chronic kidney disease stage 3, so moderate. So why is the prevalence of CKD3 so alarming? Studies from the general population have established that CKD accelerates cardiovascular aging. It causes very severe inflammation in the cardiovascular system, and it leads to remodeling and fibrosis, which presents clinically as coronary artery disease, heart failure, arrhythmia, and also sudden cardiac death. And this is also true for the aging population of people with CF. So the cornerstones of managing CKD is early detection and prevention, and it is critical to reduce the premature death from heart disease. And I want to emphasize that people with chronic kidney disease are more likely to die of cardiac complications than progression to end-stage kidney disease. So this is really critical. So the risks of CKD in people with CF are numerous, starting from the possible role of CFTR in regulating the nephron endowment, the repeated episodes of sodium and electrolyte and volume losses, which upregulate chronically the renin-angiotensin-aldosterone system, the effects of the most predominant and prevalent extrapulmonary manifestation, the CFRD, effects of inflammation and infection from various causes, as well as the medication side effects. So one of the important questions is whether there is an underlying vulnerability of the kidney in people with CF that predisposes to these insults that are being brought by the causes that I mentioned. I presented some of the data last year, and I'm going to give you an update. We are using kidneys from the CF mouse model G542X, which was characterized in 2018. This is premature stop codon with severely decreased, over 80% decrease in the CFTR protein and very minimal no function. We have 3 age groups of animals, or rather the kidneys from the animals, the young aging 6 to 11 weeks, the aging group between 31 and 38, and the old group almost 2 years of age. And I will refer to the knockout animals or refer to the study group, the G542X homozygous as knockout, and the controls with the same genetic background as wild type. So in order to evaluate the kidney insult or injury, the best place to look is the glomerulotubular junction. This is an area that is marked by the red circle, and it is where the glomerulus connects with the very early proximal tubule. It is very vulnerable because it is dependent on very high metabolic rate, and any kind of insult, including oxidative stress, is going to cause a problem and injury. The normal morphology or histology is at the GTJ is the columnar epithelium, and when injury occurs, it is transformed into squamous epithelial cells. So it makes it easy for us to actually quantify the injury at the GTJ using lotus staining. It is a special stain that is only taken up by the healthy columnar epithelial cells, but not in the squamous cells. So if you focus your attention on the green circles, so all the circles contain or actually mark the glomeruli, and the green ones mark the healthy GTJ. So you can actually see when you look at the far right in the big circle, the green circle shows glomerulus, which is round and almost like an egg cup holder, starting from the left with the gray-green discoloration, and that is actually the lotus staining. So presence of the lotus staining indicates that there is healthy tall columnar epithelium and GTJ is intact. However, in the red circle, there is just an empty or decapitated, to make it more dramatic, glomerulus that does not have the surrounding cup. So this one is injured because the squamous epithelial cells surrounding it do not take up the lotus staining. So I'm just showing the summary of the lotus negative or the injured glomeruli, and the green circles here demonstrate data from the wild-type control animals, and the red circles from the knockout animals. So even at the young age, this was the first group between 6 and 11 weeks, we saw small but significantly increased number of the injured glomerular tubular junctions, and it progressively got worse in the aging animals. So injury at the GTJ usually correlates with renal fibrosis, which occurs in the tubular interstitial compartment. So we evaluated the collagen accumulation in the wild type and control animals, and quantified the accumulation by Visiopharm software. And in this summary on the right, I am combining the data from the young and the aging animals. And as you can see, there is very discrete but statistically significant increase in the accumulation of collagen, indicating that there is some beginning of renal fibrosis at this stage. So the next set of data were obtained from the single nucleus RNA sequencing, and we have identified 16 clusters of cells corresponding to the different nephron segments, and the colors correlate the cell cluster with where the cells are present on the nephron using this schematic. So one of our first questions was if the CFTR knockout is affecting the size of the clusters. And by size of clusters, I mean the number of cells that were identified in these clusters. So you can see here that the number of cells in the proximal tubular as well as the distal convoluted cell clusters were decreased. And for the interest of time, I'm just going to focus on the proximal tubule. We subclustered the proximal tubule and noted that the decrease was present both in the convoluted as well as the straight. So these are the longest segments in the proximal tubule, but we also identified a change actually in the opposite direction in the proximal tubule cluster that does not specifically allowed us to distinguish whether they were convoluted or straight, but there were some indications from the next set of data. So here we evaluated the differential gene expression in these cell clusters, and as you can see that both increased direction and decreased direction marked here by the different colors and position were primarily affecting the proximal tubule. And I want to point out, what I want to point out is the lowest, smallest change, which is this very small change, which is the proximal tubule injured cluster. And this cluster was identified by the expression of kidney injury marker 1, KIM1, which is actually a emerging biomarker of kidney injury specifically expressed in the proximal tubule. So the expression of this marker was only present in the CFTR knockout. So that actually very nicely correlates with the fact that we are seeing some proximal tubular injury at the gene level and not in the controls. So we also evaluated the effects of the CFTR knockout on the network of protein-protein interactions, and these data were obtained from both the single nucleus RNA-seq as well as the BioGrid data bank, where we created these nodes of individual proteins with varying degrees of interactions, and the color gradient here represents the average log2 fold change across the cells in the two cell clusters, the proximal convoluted and proximal straight tubule, which were downregulated. So you can actually see that the interactions are affected in both directions in these cell clusters. And finally, we wanted to know if the changes that we were identifying at the cellular and subcellular level were also correlating with any change in the kidney weight. So this is just a very gross marker, but it may be useful. And at the youngest age in the animals where they were only about 6 to 11 weeks, we did not see any change in the weight of the kidneys, but over time in the aging and the old animals, it became significantly different with the CFTR knockout kidneys having lower weight. It has been shown already before that these animals are not growing as well as the wild-type animals. So I want to point out that there is no direct relationship between the body weight and the kidney weight. But nevertheless, it is something that we need to think more about. And the question can be answered in both ways, or question can be asked whether the decrease in kidney size indicated some chronic changes is the cause or contributes to the decreased weight. Is there some metabolic effect or the other way around? But what could account for the lower kidney weight, independent of what the cause is? So, as I mentioned before, we're born with certain nephron endowments, so we are not making any new nephrons. So the changes that are occurring in the kidney mass are related to the growth of the tubular interstitial compartment, and specifically the proximal tubule. So I'm showing data here or discussing what is happening in humans, and similar changes are happening in mice, but our size of or length of the proximal tubule at birth is only 2 millimeters, and it grows to 12 millimeters. And with that, also the interstitial compartment increases. So the changes that we are seeing in reverse during chronic kidney disease or aging are also related to the decrease in the proximal tubule size and the interstitial compartment. So the changes that we are observing with the smaller weight of the kidney are most likely related to the changes in the proximal tubule. So what we have learned so far, the data that I showed you showed that at baseline, CFTR knockout negatively affects the proximal tubule. And this was shown by the injury to the glomerulotubular junction, the increase in renal fibrosis, decreased number of cells in the proximal tubule clusters, and also increased number of injured cells marked by the KIM1 expression, as well as altered gene expression in differentially affected gene expression and altered protein interactions, as well as decreased kidney weight. So is there something that we can translate to the clinics? Our data suggests that people with CF are predisposed to the maladaptive responses to superimposed renal insults, and that may precipitate and increase the risk of chronic kidney disease. And being aware of the vulnerability to chronic kidney disease may help to reduce the risk of premature death from cardiovascular complications in the aging population with CF and improve outcomes. So I just want to thank everybody who contributed to this program, and I also want to specifically acknowledge the funding from the CF Foundation for the multi-PI grant that we are moving forward to provide more and more data on the role of kidney in people with CF. Thank you.
Thank you very much for that very interesting talk. Please don't hesitate to speak up if you have a question. Yeah, there's no mic, so I do want to remind that there is an app, so if you feel more comfortable submitting your question through the app, please do so.
Is it known that the stem cells that regenerate the glomerulus, how they fail in this chronic kidney disease, at least in the mouse? You know, like why is it that fibrosis occurs and they lose their?
We don't know. That's a good question. Generally, we don't regenerate the nephrons glomeruli. There is probably some regeneration in the kidney tubules, but that's a really great question. I don't have the answer.
I have a question. Have you attempted to quantify nephron numbers in, say, neonatal mice, and is there a difference?
Yes.
Or is it something that happens later as the animal ages?
So we haven't done it. This is a plan. We have to actually do these studies in live animals. So we're preparing for those studies. These are just kidneys that were sent to us after they were the animals were sacrificed. So we cannot do that. It has to be done in vivo, but there are several good models and methods for doing it.
And if you were to induce inflammation at some other site, would you predict that that would exacerbate this process?
So, well, this will be my prediction, but also a good validation. So, again, these data are just showing the very baseline situation without any additional stress, but using additional models for kidney injury, both related to specifically for the risks that are seen in people with CF, as well as just general kidney injury models, will definitely add some more information.
Great. There's a question from the app. Is there evidence of lower GFR or accumulation of waste products like urea?
In mice.
CF, in mice. I
In
guess.
mice. We don't have the data yet, but we're gearing towards getting those. Mm-hmm.
Yes, hi. Very nice talk. So just a quick question, because I'm not familiar with that model. Those mice do they have any form of diabetes or not diabetic at all?
Diabetes, that would be a question for Craig. I don't think so, but, right, so yeah.
And that doesn't look like the, okay.
Saskatchewan, I noticed that you didn't include any CFTR dysfunction in the tubule itself as a cause for kidney disease. So the lack of CFTR function of beta cells don't contribute to kidney disease?
So if I understand your question correctly, you were asking about some other dysfunction in the kidney tubules related to CFTR, yes. Yeah.
But I understand that beta cells in the collecting duct express
Yes.
CFTR.
Yes.
So you don't include that as a cause for your...
Well, again, for the length of the time that I had here, I focused specifically on the proximal tubule and the potential effect, the contribution to chronic kidney disease. But there are additional data on the effects of lack of CFTR or the common ancestry of some cells in the distal tubule with the ionocyte. So. Data coming from the ferret model. So there are lots of other potential effects. So this is just focusing on what we were able to get from the histology and focusing on the proximal tubule. But that's a really great question, yes.
Great talk, Julie from the University of Iowa. Are there any data for the patients with CFTR carriers or people with CFTR mutations, like homologous patients from
So those are really great questions, and I hope that we are going to be able to study in more granular fashion. There was not really much of a focus to study kidney in people with CF because the priority was to prevent mortality. And I think now the time is, and especially with the ability of correlate or study the effects with and without modulators. I think we're going to be able to detect and identify the changes that are happening and are secondary just to the CFTR dysfunction, whether this is the primary risk or the effects of other metabolic changes that are happening as a secondary effect. So the field is, I think the kidney will provide a lot of good information, both from the basic research standpoint, but also for the patients, for the future care and prevention of CKD.
Thank you very
Okay,
much.
thank you.
Thank you.
And our next speaker is Dr. John Engelhardt.
For those of you that weren't here earlier for the introduction, you can tell I'm not yelling "ye." I'm honored to give the presentation for her and to thank her for feeding me the great data and slides, and thank the organizers for allowing us the opportunity to present our work. So these are my disclosures. So I have relatively few introductory slides. I want to introduce to the pancreas where is CFTR expressed, and it's expressed within, let's see, we got a pointer, okay. Expressed within the ducts, and where it plays an important role in regulating pH of the duct to keep the pH high so the enzymes are not activated prior to moving out into the intestine. CFTR is probably also expressed in a central acinar cell, a very specialized ductal cell, and a subset of acinar cells based on single-cell RNA-seq. We're lucky to have a number of larger animal models, ferrets and pigs, that have cystic fibrosis that develop pancreatic disease that you see on the bottom and lung disease you see on the top. This involves obstruction of the major ducts, degeneration of the acini, and then adipogenesis that occurs later during the process. And mice do not get pancreatic disease, and it's thought to be due to expression of an alternative chloride channel. And when I present data from the ferret on diabetes in the pancreas, I like to present some of the early work and collaborative studies that went on to humans with Katie Larsenodi, who's in the audience. And so cystic fibrosis in CFTR knockout ferrets, the pancreas disease is extremely synchronized. You can see here, this is taking blood prior to, all right, I'm going to have to switch pointers, I guess, prior to feeding, then one after feeding at birth. And ferrets at this stage have no fecal elastase at birth, so they have a very rapid and synchronized acidic degeneration. That's accompanied by fibrosis, inflammation, and then adipogenesis at the ages that you see there. And it's extremely synchronized in the way that you can see here on the right, we have mapped RNA expression as a fibrotic index and inflammatory index, the sum of these mRNA cytokines and adipokines. And you can see that there's a glycemic crisis at about 1 to 2 months that animals are spontaneous glycemic. And then a very rapid recovery. And I'm not going to talk about this, but that recovery involves the regeneration of islets that have been destroyed during the early phase of acinar degradation. And so exocrine pancreas disease in the CFTR knockout ferret is very rapid. And we were interested in understanding how age of onset of pancreatitis within the model affects progression to CFRD and the glycemic abnormalities. So one of the models that we generated very early was a G551D knock-in. These animals have, you can look at fecal elastase here on the right, in which animals that are homozygous maintain normal fecal elastase, they're pancreatic sufficient. Animals that are heterozygous become pancreatic insufficient at about 1 to 2 months of age, and these are the body weights on the left. So the animals shown by the arrows here have to go on pancreatic enzymes, so they're going to lose weight and would have to be sacrificed. And you can look at the morphology of these various animals here, comparing a knockout, is at an adult stage, to wild type, to homozygous G551D with normal on VX770 or ivacaftor, the CFTR modulator, heterozygote animals here next to the right, and then homozygous animals that are taken off VX770 that then go on to develop disease. If one compares this to human, you would ask, well, the heterozygote
carriers of CF are normal. So why do the heterozygote CF animals become pancreatic insufficient? And well, it has to do with the way that we originally made the first G551D knock-in. It was made by somatic cell nuclear transfer, and we had to target the exon mutation, and we left a neomycin cassette in the center that had its own promoter and so forth. So down on the lower left, you see the CFTR mRNA ratio to HPRT, and homozygous animals have 50% of CFTR mRNA compared to a wild-type animal. So as it turns out, if you have 25% CFTR mRNA in green, you see that you become pancreatic sufficient. That's not enough, at least in a ferret, to maintain pancreatic sufficiency. So we remade the model, a clean knock-in using new technologies, CRISPR-Cas9, that we didn't leave a fingerprint in the intron that would affect expression. And these animals have normal pancreatic enzymes. I don't have the heterozygote mapped here, but they have normal levels as well. So to get back to this is what we call the hypomorphic model. So this is the one that I just showed you the fecal elastase data for. You can see here that these are CFTR knockout. These are heterozygote. You can see that they have glucose. This before feeding and after fast, and 1 hour after feeding. They have abnormal glucose tolerance, or postprandial glucose tolerance. And you can see here these two outliers, the pancreatic insufficient in this group, the homozygous, and here, and basically they map to where you would expect to have normal glucose after feeding. This is just quantifying this by a mixed meal tolerance test, a formal one where you can see the heterozygote animals basically look CFTR knockout, this area under the curve, and just reminding you that the pancreatic insufficient ones decline over time, but the point of why I'm showing this is you can be CFTR knockout, or you can decline in the neonatal period, and your outcome for glucose intolerance is basically the same. So we went on to try and do this in a bit of a more formal way. Now we've turned to the clean knock-in. So we know we have 100% expression. We get about 75% rescue on a CFTR modulator, VX770, and so we have what I'm calling here an early group that goes off VX770 at 1 month and a late group that goes off at 4 months, asking the question whether or not the outcomes, glycemic outcomes in these animals are different. And if you, this is again measuring glucose at fast and 1 hour post feeding, and you can see in the younger group, the one that came off at a month, it looks like a CFTR knockout. It has this crisis period, the equivalent of about 1 to 2 months off of VX770, and you see in the postprandial this very similar thing. What is interesting is that in the laid off group, you can see here that the glucose postprandial feeding is getting upwards of about 200, which is abnormal. This is at 100, or this is at 60 minutes, 1 hour post feeding. And on the right, you can see that in a formal mixed meal or oral glucose tolerance test, so this is at equivalent stage or going off of VX770 in the two age groups. And the one that went off later had worse glycemic outcomes. This would be classified as CFRD. And the one that went off at the earlier time point would be indeterminate glycemia, a less severe state. And this just classifies the two, the number of animals or the percent of animals that are seen in each of these groups. So going off the CFTR modulator later in life led to worse outcome, potentially because the pancreas has the ability to regenerate and remodel better when it's younger. So 1 to 2 month old ferrets are physiologically similar to about a 2 to 4 year old toddler, and I based this on Katie Larson-Odie's study with Yaling that demonstrated this glycemic crisis occurs in both those age groups compared to very young ferrets. And the onset of C. F. pancreatitis impacts longer term glucose tolerance, with it being worse if you go off later. So we wanted to ask, what are the age-dependent attributes that might control longer-term glycemic status, and what's the regenerative capacity of the pancreas using a number of different ways in the CFTR modulators? And so here are some studies where we took advantage of the fact that we're trying to age animals by taking them off at 3 months to get lung disease later in life. This tends to be technically, from a rearing standpoint, the easiest time to take a ferret off modulators without having secondary complications. And then we had animals that we kept on for life, males typically that were breeding and retained fertility. So, and just to remind everybody, ferrets wean at about 8 weeks of age. So you can see here, animals on, pancreas looks pretty healthy. Here is the residual pancreas of animal that went off at adulthood. And if one looks at the glycemic outcomes here, this was an oral glucose tolerance test comparing animals that were on VX70 for life, much better glucose tolerance. This is area under the curve compared to ones that came off at 3 months. Again, these are all adults. And then fecal elastase, obviously, the ones that came off don't have fecal elastase, don't have acinar cells anymore. So then, using an alternative approach, we took animals off that were earlier at 1 month of age, again, weaning at about 8 weeks. And comparing these two groups led to some interesting findings. So we can see here, this is the group that's on, this is group 1. You can see, and we're measuring right at about this time, about the time of weaning. There are a subset of animals that would be considered pancreatic insufficient. Obviously, the animals that came off at this time are going to be losing their acinar mass and become pancreatic insufficient. And indeed, they remain pancreatic insufficient, go down to about 0, like absolutely no fecal elastase. What was interesting is this group here was transiently pancreatic insufficient across the weaning period. They came back as adults and they all became pancreatic sufficient, very significant. So what's the take home for this? Well, the CFTR modulators are not 100% protective of the pancreas, at least in a ferret. You can see here, these are 50 and 100 days, so animals are weaned right in between here, probably considerable stress on the pancreas. This is starting to eat their own food, and you can see some fibrosis and acinar degeneration in pockets around here. But by the time they're adults, pancreas looks pretty normal. So there's evidence of pancreatic regeneration. I'm not going to talk about this, but we're trying to find the stem cells that are responsible for this using lineage tracing in the ferret. And I think it's an opportunity for attempting cell-based therapies or gene editing therapies if there's a selective advantage to the cells that correct CFTR. So removal of live calf after 1 month G551D ferrets leads to better glycemic outcomes than if they come off at 4 months. And 29% of if you consider CF ferrets at that age, toddler age, become pancreatic insufficient, but they're rescued later in life. So we hypothesize that the pancreas of these neonatal toddlers in CF ferrets have a greater regenerative capacity than adult or juvenile or adult ferret pancreas. And so we haven't completed these studies, but this is the first group that's been taken off at a younger age, 1 to 2 months here, and then back on to VX770. And you can see here from a growth standpoint, pancreas of animals on here before they go off, pancreas looks great, very healthy. You can see these pockets of cystic looking areas when they're off VX770 after a month, and then the pancreas regenerates back. So what does it look like from the standpoint of pancreatic enzymes? Well, you'd expect these are all the animals that were in the study. We've classed them into 2. These are ones that stayed pancreatic insufficient after they went back on, but they all go down at 2 months as you'd expect, and then coming back up, acinar mass regenerates and they're secreting fecal elastase. And if one classifies them into the various groups here, we got animals with no fecal elastase here in blue, a larger proportion of pancreatic insufficient ferrets, and slowly the pancreatic sufficient grows and may increase even further. This is another way to look at it, but also comparing the fecal elastase to the glucose tolerance. And you can see that the ones that had the hardest time coming back to be pancreatic insufficient are the ones that have the worst glucose at 120 minutes, although it's not diabetic level here. And this is just an alternative way of looking at that. I think larger dots have more fecal elastase here, and this is just looking at the oral glucose tolerance from the standpoint of the peak glucose. So that would be here at 60 minutes, and then area under the curve. And you can see significant correlation with between abnormal glucose tolerance and fecal elastase levels. If one then compares this to the earlier group that went off at 1 month and stayed off, again, one sees that the off group has significantly worse glucose tolerance compared to the on group, and this is just area under the curve showing the same thing. And the ongoing studies that we're doing now are to go into adult animals and take them off for a month and then ask what's their probability or likelihood of regaining pancreatic sufficiency, and how is that linked to their glycemic status when they're done. So I'll just, 2 more slides and then conclude. So an interesting area about pancreatic regeneration of the acinar cells, and there are hints of this in humans, and it's thought that acinar cells, when they degenerate, form a ductal-like cell, so this would be acinar ductal metaplasia, that can then regenerate the acinar cell mass. And indeed, if one looks, this is RNA in situ hybridization of markers of acinar cells. This is RNase 1. You can see these ducts here in CF animals, this CFTR knockout, that are assuming this acinar identity. This is at 2 months, where it appears that this gene is expressed in central acinar cells more than likely, but one still sees this expression within the ducts. And amylase B, which is another acinar cell marker, is also expressed within the ducts. So to conclude, in homozygous G551D ferrets, ivacaftor is not fully protective, but the pancreas has an ability to regenerate. This is greater in younger animals, at least from what we've seen so far. And acquiring pancreatic insufficiency later in life appears to give worse outcomes from a glucose tolerance standpoint. So we really we still have a lot to learn about this and the stem cell compartments that are mediating this, but there's hope that there may be other ways other than the CFTR modulators to promote this regenerative process. So I'd like to thank those collaborators, both within the larger lab, but Yaling Yi head up all this work, and faculty collaborators, as well as Chinzen Sun and Ziying Yan. Thank you.
I'm wondering if we're going to look at the linear transformation possibility, the regeneration period, because there may be an increase in the reservoir linear transformation.
Yeah. It's fascinating. So the question was, is there an analogy to malignant transformation in the ducts that's going on? There's an incredible similarity to how the ductal cell fate, what that acquires in a CFTR knockout animal compared to pancreatic cancers that emerge from pancreatitis. We have a paper coming out, I think it's in iScience, that talks about the phenotype of this ductal cell, but it activates many of the factors like PDX1, which is a very early progenitor pioneer factor you might consider that controls pancreatic formation. But a really interesting question. Yeah.
John, I have a thought. I was staring at the data thinking about the whole importance of toddler and feeding as a time point. Have you any thought of looking at basically teenage, like kind of around ability to start reproducing age?
That study right there is about, actually that's just before, 6 to 6.5 months is when fertility, it's very close to that. So that would probably be the equivalent of about 13 in a female, and somewhere in there.
From a human perspective too. And also that's the time that humans are most likely to discontinue their correction care.
Yeah.
Against the wishes of their physicians. So that would be a really interesting.
What's amazing to me is that, pancreatitis is supposed to be extremely painful. Even in the older animals, when you take them off VX770, it's not like they're hunched over and not moving around in the cage. There's something different about, losing CFTR function and how that emerges, the pancreatitis, but also destruction of the acinar cells. I don't know if it involves like cutting the nerves. Andy's studying some of that, but that makes it less painful than if like other forms of pancreatitis.
Thank you for the great talk. Our next speaker is Dr. Patricia Scott.
Hello, and thank you for the invitation to speak about our work. Okay, so let's see. how does it go? Okay, so no disclosures. So this project is part of our ongoing work to look at the relationship or to look at the effect of loss of CFTR on development of colorectal cancer. And it's well established that the loss of CFTR increases the risk and severity of colorectal cancer in people with CF. And more recent studies are now showing increased risk in CF carriers and in CRC patients who have low expression of CFTR in their tumors. However, we still don't have a good understanding of the biological connection between loss of CFTR and colorectal cancer. So in our group, we're looking at the crypt microbiome as a possible factor that is involved in this connection. And so why the crypt, CFTR is expressed in the intestinal crypt, and intestinal stem and progenitor cells, which are the cancer, thought to be the cancer-initiating cells in colorectal cancer, are also located there. So loss of CFTR has the potential to directly impact the environment of these likely cancer-initiating cells. And so why the microbiome, a GI manifestation of colorectal cancer is intestinal dysbiosis of the intestinal microbiome. And in recent studies, it's now being shown that there's overlap between the changes that occur in the intestinal CF intestinal microbiome and those changes that occur during colorectal cancer. So putting all this together, our model that we're trying to test is that loss of CFTR in the crypt causes changes to the crypt microbiota. This then causes change to stem and progenitor cells, and this contributes to tumorigenesis. To look at this, to begin to look at this model, to test this model, we used an intestinal-specific CFTR knockout mouse model, and we also looked at normal non-tumor colon tissues. So we first asked the question of whether the crypt microbiota of CFTR knockout colon differs from that of the wild type. And to do this, we isolated colon crypts and fecal samples and used 16S rRNA amplicon sequencing to identify the bacteria species that were specific to each sample. And then in second objective, our question was whether bacteria that are differentially abundant in the CFTR knockout crypt cause pro-oncogenic changes. And for this, we used, our readout was outgrowth of colon organoids. And so to explain that, outgrowth depends on the number and fitness of stem cells in the original population, and also the favorability of the experimental environment to the growth of those stem cells and expansion of those stem cells. And so since many colorectal cancers are thought to arise from stem cells, from intestinal stem cells, factors that affect that promote outgrowth have the potential to be oncogenic. So, let's see. Okay, so going back to our first objective, we first compared the composition of the colon crypt microbiota to the fecal microbiota. And so looking on the left, I'm not going to even try to do the pointer, looking at the principal coordinates analysis on the left, we see a clear separation between the crypt samples and the fecal samples, and this indicates that these populations differ in bacterial species composition. And this is supported by analysis of the 16S RNA data, ribosomal RNA data, which shows when the 10 most abundant taxa, the are analyzed and the relative abundance is analyzed, we again see a very distinct difference in the composition of the crypt and fecal samples. And so these results support that or give some support to the idea that our crypt isolation was effective. So the next thing we looked at was a comparison that was critical, basically, to this study, and that was to look at to compare to analyze the 16S RNA data to determine if there was differential abundance of any of the taxa. And so looking at the 10 most abundant taxa in these populations, we found that only 2 of them differed in abundance. And these were an significantly increased abundance of lactobacillus and of bifidobacterium. And so an important feature of this is that both of these bacteria, their major metabolite is lactate. And so considering that recent studies have shown that lactate is bacterial lactate from lactate acid bacteria, bacterial lactate is able to promote the expansion of LGR5 stem cells, and also it's pretty well established that tumor lactate that arises from tumors is considered an oncometabolite. So based on this, we think that it's possible that this increased abundance of these lactic acid bacteria in the crypt might be contributing to the development of colorectal cancer. So to follow up on this, we looked to see if the increased abundance of these bacteria actually had an impact on the exposure of the intestine to lactate. Okay, so we approached this in 2 different ways. And so in the first analysis, Josh Fletcher carried out an analysis of the 16S RNA sequence using a bioinformatics program known as PyCrust2 that is able to predict the biochemical pathways that are enriched in a population of bacteria. And so when he did this, he found enrichment for two pathways that are involved in lactate synthesis, the heterolactic fermentation pathway and the bifidobacterium shunt. And so then Rahul Bhattacharya took a different approach, okay, and looked at whether or not there was evidence of increased lactate in the host tissue itself. And so he compared colon tissue, which is exposed to microbes, it's exposed to commensal bacteria, and he compared that to organoid RNA, or to organoids which are not exposed, they're grown in sterile medium. And so he looked at the enrichment of lactate metabolism-associated gene signatures, and he found that in the CFTR knockout colon tissue, there was, in fact, enrichment of those lactate-associated signatures, but in the organoid RNA, there was not. So taking those together, we considered that there's evidence for post exposure to lactate, and so we next asked what was the effect of lactate on the intestinal crypt. And so, let's see, so I'm running out of time. Okay, so for these experiments, colon organoids were dissociated to single cells. Equal numbers of cells were plated, and they were grown out for 5 days. And at the end of 5 days, the images were counted to determine the size and the area and the number of organoids. So in the first experiment, we looked at the effect of CFTR knockout on organoid outgrowth. And what we see is that there was an increase, let's see, does this work? There we go. An increase in the total number of organoids greater than 50 micrometers, and also that increase was significant in the larger organoids greater than 100 micrometers. So we next look at whether lactate promoted outgrowth of lactate by itself promoted outgrowth of CFTR wild type organoids. And again, in this case, we're looking at the ratio of the number of organoids in lactate-treated organoids versus vehicle. Okay, so again, we see an increase in the total number of organoids, and we see that increase occurs in the larger organoids greater than 100 micrometers. So then we looked at the combination of the effect of lactate exposure plus loss of CFTR. And so focusing just on the organoids, CFTR knockout organoids that were treated with lactate, okay, we see that these organoids, that there was more organoid outgrowth in these organoids than in the CFTR wild type or in the CFTR knockout organoids that were not treated with lactate. So lactate had an effect over and above the effect of knockout. And even though both the wild type and the knockout organoids were treated with the same concentration of lactate, okay, we saw that, we saw that there was, that the CFTR knockout organoids treated with lactate showed more outgrowth than those, than the CFTR wild type organoids treated with lactate. So putting all three of these together, okay, we see that CFTR knockout, loss of CFTR and lactate exposure both caused an increase in outgrowth. And considering that outgrowth reflects the stem cell number and fitness, we then propose, or we think that possibly this effect is actually affecting the stem cells. So to look at this in another way, Josh Fletcher made CFTR depleted CACO2 cell lines and using shRNA targeting, and he analyzed the expression of several genes in the Wnt beta-catenin pathway. And the Wnt beta-catenin pathway is central to maintaining normal stem cells and also is the pathway that's most commonly dysregulated in colorectal cancer. So looking at the expression of three genes in this pathway, CTNNB1, LGR5, and AXIN2, he had two findings. First of all, that depletion of CFTR caused an increased expression of these three genes. And further, then addition of lactate caused an additional increase in the expression of CTNNB1 and LGR5. So that was consistent with our organoid results. But there was also one result that was kind of unexpected, and that involved the CFTR. So let's see. Okay, so looking at the CFTR, in which CFTR has been depleted, or the samples in which CFTR is depleted, we see a decrease in expression as we expect. But looking at samples in which CFTR was not depleted with short hairpin RNA, we see that lactate caused a really strong decrease in the expression of CFTR. And so we actually don't know what to make of this, but we think it could be important because it could be a way that that CFTR expression is decreased in people with low expression of CFTR in their colorectal cancers. Okay, so let's see. Okay, future work. Let's see, so I'm going to skip that, okay, and I'm just going to end with the acknowledgments of the people who worked on this over the years, and acknowledgement of the agencies that have funded the work. So
Any questions?
So the villain promoter they use to conditionally knock out CFTR just in the gut, is it expressed in the crypt?
It's villain?
The villain?
Yeah. Yes, yes.
It's expressed in the entire epithelium of the intestine? Okay.
Actually, I was very I'm granular expert in that, and I was wondering whether lactate-producing bacteria, which are usually supposed to be good for your gut, the microbacteria, would then actually be something negative if you.
Right, and that's the reason why, yes. So that's our question too. So yes, we've shown that there's this organoid outgrowth, but that organoid outgrowth can actually be beneficial if you're in a situation where you need to replenish your epithelium. So what we think is that it might be a double-edged sword, and where when you lose CFTR, you cause a big increase, okay, in this bacteria, a big increase in lactate, and then that kind of pushes things over the edge so that it's dysregulated and causes too much growth.
And the other question is more definition question. Would you consider in your context CFTR to be a tumor suppressor?
Yes.
Yeah. Thank you.
I have one question. Have you ever looked at the CFTR function
In
organoid?
We haven't, but I don't know if Blaine Clark is here. Other people have, yes.
Okay.
It's been confirmed, yes.
Okay. Thank you.
Okay.
Thank you for the great talk. Our next speaker is Ted Nett. Thank you.
Sure.
All right, thank you for the introduction, and thank you for the opportunity to speak. I am a graduate student at the University of Iowa studying under Dr. Ling Yang, and just grateful to speak to the audience as a student. I will be over at poster 385 if anybody wants to come speak after this talk, just after the session. Today, speaking on the role of the micropeptide BNLN in the context of CFRD. No disclosures. So starting broadly, we are novices to the cystic fibrosis world and came in from a more traditional diabetes standpoint. And I want to start with kind of introducing the concept of link RNAs and micropeptides. So there are several regulatory RNAs that do not produce proteins, among them microRNAs and then these long non-coding RNAs or link RNAs. Several of them have been implicated in the development and progression of several diseases, including type 2 diabetes, through effects including beta cell development, apoptosis, insulin production, and then outside of the beta cell in insulin resistance. Over the last several years, many of these thought to be long non-coding RNAs have actually been found to encode for what are called micropeptides, small proteins that do carry out a range of functions. Some of these more prominent functions include regulation of calcium in muscle, maintaining metabolic homeostasis, developing muscle, and regulating ER stress. At the time that our group entered into this project and before I joined the lab, there were no known micropeptides acting in the beta cell. So we went in search of them. And using computational methods, tracing essentially the RNA abundance and the coding potential of those RNAs, we came across a gene called TUNAR, which in fact encodes for a protein we named beta cell and neural cell regulin, or BNLN, shown here. Small peptide, and most prominent feature I'll show here is the highly conserved hydrophobic region that we presume to be a ER membrane-associated protein. I'm going to limit how much of the characterization we did in this early work. This is published. Some of it is out at the poster session. But for the purposes of this talk, I'm going to just introduce the insulin secretion support that we identified in this paper. So if we isolate islets from regular diet mice, we find that, and supplement them with BNLN or a control vector, we find a significant increase in insulin secretion in the BNLN supplemented islets. Taking a high-fat diet mouse, we isolated those islets again, checked for gene expression for TUNAR, and found it significantly suppressed by about half. Interestingly, and this is driving a lot of our next questions after this project was wrapped up, we found that if we took these high-fat diet islets, which are barely responsive to glucose as it relates to insulin secretion, and supplement BNLN to those islets, we're able to restore them to almost normal levels of insulin secretion. And critically, these effects do persist in islets from human donors. So at this point, I enter into the project, and for this paper, we were relying on tagged constructs for this new protein to observe it. Since then, we have developed an antibody against BNLN, and one of my early tasks was to look into where is this thing expressed endogenously in the pancreas. So we started looking for different disease applications. Obviously, we did some obesity, high-fat diet work, and due in part to availability and to remain in the diabetes world, we looked first at type 1 diabetes. We had the NOD mouse model on hand. Sectioned some pancreas stained with BNLN, which is shown here in red, and insulin in green to identify the beta cells. What I want to point out is that while there is, do I have a pointer? Is that, we're going to find this little thing. Get myself in trouble up here. So while there is some co-expression shown here especially, you can see the yellow markings in there between BNLN and the beta cell, I want to highlight over here in this type 1 diabetic mouse this really, really dense population of red BNLN expression that is not associated with insulin expression or beta cell. So that is some non-beta cell type that we have yet to identify at this stage that is highly, highly expressed in the disease condition. We were also looking in the search for other applications and looking through sequencing data. This is how we come into the cystic fibrosis world. We found that in cystic fibrosis human islets, we found a similar suppression of this TUNR expression that we saw in the high-fat diet islets in our published work, which led us to believe, okay, if we see a similar defect in TUNR BNLN in this disease, perhaps we could go in and supplement and see a similar recovery that we would see in our high-fat diet mice. Here I'm showing some single cell. On the left, you can see different, this is for pancreatic tissues. So on the left is the different cell type clusters. On the right is the relative expression of TUNR throughout those clusters. I've highlighted the beta cell population, but you'll also notice significant expression in alpha cells, delta cells, and ductal cells as well. So this is expressed at least in part throughout the pancreas. So some broad questions as we're entering into this project, and I'll preface myself and say a lot of these are unanswered for the purposes of this workshop. First and foremost, what is the spatial temporal expression of BNLN during CFRD throughout the phases that it progresses through, as Dr. Engelhardt explained earlier? Second, what is modulating BNLN expression and function? And third, more broadly, is what is the role of micropeptides like BNLN in beta cell dysfunction in CFRD? So even if it's not BNLN specifically, could what we learned about BNLN tell us something about this class of proteins that is just becoming more known in the last several years? Also, as Dr. Engelhardt mentioned, the mouse model is not especially useful for studying the pancreas, which was not great in our mouse lab. So due to the generosity of the Engelhardt lab, we were able to get access to the ferret knockout model of CF. And here I'm showing a wild type ferret. This is in the age for the phase 2, or that initial loss of beta cell mass and loss of glycemic control. You'll note some BNLN expression pattern throughout the cells, including in the beta cells and in the non-beta cells. And if we look at our CF animal, you'll see very, very similar patterning to what we saw in the type 1 diabetic, with that distribution throughout, but a very, very strong cluster not associated with the beta cell. Really wish we knew what that was. That is a high priority. So there is a similar, what I can say here is there's a similar linkage between these multiple forms of diabetes, and BNLN may be performing a similar role within the islet, if not the beta cell, or there's communication within the islet that is affecting beta cell function. Being that our focus is diabetes, my methodology early on in this research was to stain for insulin and then find the insulin and take the pictures of what was happening around the insulin. Over the course of this project, that has become less feasible because I was finding an odd trend where there was extremely heightened BNLN expression in these animals around the pancreatic ducts, which was not necessarily what we expected from our early characterization. Before we dove in too far to identify what this possible role could be, I wanted to validate that it is at least present in humans as well to see whether it was worth pursuing, which we also don't have great access to. So thanks to a new collaboration with the Amy Ryan group at University of Iowa, they're developing pancreatic organoids from human donors that are iPSC derived. And we were able to get access to some of those and stained, and we do indeed find endogenous BNLN expression in these human iPSC-derived tissues. So now I got to figure out what in the world it's doing in the duct. And that is something I would love to talk about people here with if you have any clue or guesses that I could start looking into. To this end, TUNAR, before it was known to encode for any peptides at all, was identified as a factor in beta cell proliferation, as known as functioning as a long non-coding RNA. And one of the things we're trying to sort out now is whether the protein BNLN is actually carrying out that function, or whether it is, in fact, the RNA still carrying out its capacity in a long non-coding RNA that still encodes type way. So I'm pulling this from a Balboa paper in Nature Biotechnology, just looking through sequencing, and up here showing essentially the process to go from a stem cell to mature islet organoids that their group carried out. And in looking through their sequencing, we were interested to find that in the early stem cell, but destined to end up as a beta cell fate, we see heightened TUNAR compared to what we see in the adult beta cell. So perhaps there is something here that is leading to beta cell proliferation as a protein. And this is of interest to the third phase of CF, in which we see a regeneration of beta cell mass, a beta cell mass within the islets. So this is incredibly preliminary. This is one sample each, but again, through the Ryan group, we're beginning to investigate this through their iPSC model. As of right now, we only have these first stages up through iPSC to the pancreatic progenitor stage, looking at both iPSCs derived from healthy donors and from donors with CF. Now, super preliminary. Trending toward, as we're moving into pancreatic development, we're seeing an increased expression of TUNAR in these iPSC cells. Next goals would be, first of all, to get additional samples to verify that this trend is persisting, and then to move into more mature cell types, including the ductal cells and the endocrine cells that we're more interested in. Last thing I want to touch on here is that question of what is actually driving changes in BNLN and TUNAR expression. So we know that these different disease conditions are manipulating it, but it can't be as simple as CF exists, therefore BNLN changes. There's something present in the CF pancreas that is manipulating this. Here I'm showing the one that I am most confident in, and we are investigating a few others, but we found a pretty strong correlation between an acute glucose challenge and an increase in this BNLN expression, or TUNAR expression, excuse me. So as quickly as 1 hour after a high glucose challenge or a stimulatory level of glucose, we see a significant increase in TUNAR, that trend beginning as early as 30 minutes after exposure to that challenge. In a frustrating and confounding result, this does not persist when we do a longer-term culture. So for this, we took mouse islets and cultured in that stimulatory glucose for 10 days and see absolutely no difference in TUNAR expression there. So this may be a relatively short-term stress response that is not able to persist, and that might explain some of the differences that we see in that middle phase CF versus the gene expression data that we're seeing from these later stage disease, both donors and animals. So limited conclusions in a sea of future directions. BNLN is highly expressed in select islet cells in both CF and other forms of diabetes. BNLN is endogenously expressed in the pancreatic ducts and may play important roles in pancreatic development. And TUNAR expression in beta cells is regulated by acute but not chronic glucose exposure. Great thanks to the members of my lab, in particular Mark Lee, who led off the early work in this that was published, and our collaborators, with special thanks to the Engelhardt and Ryan groups for giving us the models to reasonably study these questions. Our institutional support and funding support as well, including the CF Foundation. Thank you all, and happy to take any questions.
So, at least in cystic fibrosis ferret islets, they secrete more of their insulin at baseline, so
right.
Low glucose, and they have less insulin reserve, probably because they're stressed.
Sure.
So that comes along with, there's a higher basal calcium level in an islet that's probably responsible for that increased insulin secretion.
Right.
So BNLN.
Forgive me for the acronym.
ER, right?
Correct.
Right.
Yep, I didn't get into that here, but we did find in that initial paper a close interaction, glucose dependent with circa-3. We believe it's suppressing circa-3 to maintain high cytosolic calcium levels.
Okay, so it was up in CF, that would make sense.
Correct. Yep.
Yeah, but that's maybe different than treating, that may be part of the problem, treating with the peptide.
Sure.
That if you're stressing that, the beta cell will secrete more.
Yeah, that makes a lot of sense. We have not done the treatment directly on CF samples, just due to the lack of isolated ferret islets, but that would be an interesting way to test that specific mechanism, to test that between our models and see whether there is that difference there.
Yeah, it'd be useful to have a way that you can turn it off.
Oh, right, yeah, absolutely.
Have you ever done the turn off in the mouse side?
So we are a couple weeks away from our knockout mouse in a beta cell specific. So, yep, that'll hopefully by committee meeting, that'll be ready to go.
I want to follow up a little bit on the knockout mouse.
Sure. Yep.
One of the very first slide, I think you showed, you don't have to go back to it. You
Okay.
just showed sequence
Oh,
homology
no.
between different sequences. And it looked to me,
Yep.
correct me if I'm wrong, it looked to me that the protein coding region of this long non-coding RNA is more conserved than the other regions of the molecule. Is that correct?
Yep.
So,
Yep.
I'm wondering if that can shed light on like what the function of this long non-coding RNA dynamic, and the dynamic between the RNA and the protein.
Right.
And how you're planning on, animal model and movie, how you're planning on knocking it out.
Sure. So our current knockout model is just a tissue-specific LOX-CRE, and so not the full RNA. The full RNA is about a 50 kilobase, and this guy is not anything close to that. So we're taking out just the second exon that is encoding for this, and it's a fraction of the full RNA. We've done in that published study a construct that is just removing this second start codon, which actually initiates the production of this protein, and tested the RNA effects, and that is not supporting the insulin secretion. As far as this highly conserved region, we are considering putting in a scrambled version that we could also treat with. Because looking at this, they're a relatively basic hydrophobic region, and if it is the transmembrane portion, keeping it in the ER, that should be able to fit with about anything. But with it being so highly conserved, we do wonder if that is more functional than just keeping it stuck in place.
Okay, thank you for the
Thank you all.
So our last speaker is the Oscar Omar Morales.
Hi, everyone. My name is Oscar Omar Morales. I'm coming from the University of Saskatchewan, and I'm going to talk about this novel in vitro system to study the gut-brain axis using enteroids and sensory neurons. There are no relationships to disclose related to this presentation. Okay, so gastrointestinal manifestations in cystic fibrosis are extremely common and have recently become a research focus. These manifestations include stomach cramps, constipation, heartburn, bloating, lack of appetite, nausea, among others. Some of these GI luminal complications in cystic fibrosis have been considered secondary to the decrease in CFTR function in epithelial cells. These malfunctions include fat malabsorption, pancreatic insufficiency, gut hormone dysregulations, bacterial overgrowth, dysbiosis, and gut inflammations. Additionally to this, the CFTR might have a direct impact in abnormal contraction of smooth muscle, but our project focused on the alterations in intestinal mesenteric ganglia. This is because the motility and secretions are controlled by the enteric nervous system, but this is modulated by the sensory neurons that innervate the gut. So we are interested to study the brain-gut axis with these neurons. This is because preliminary data from our lab shows that the sensory neurons in CF are disrupted in a model of swine of CF. Here I can show you that the current measured by the CF neurons is lower compared with the wild type, and it's significantly different. Also, these neurons had disrupted the chloride homeostasis, which is necessary for the excitability of the neuron. In addition to this, we have that the chloride transporter, NKCC1, along with other proteins related to the CFTR pathway, like Wink1 and OSERC1, are downregulated in the CF model. So with all of this, we are interested in study this brain-gut axis. For this, we isolate from the small intestine in the proximal portion of the jejunum, we isolate the crypts and we seed it intramatricial to develop these organoids. Here I show you how these organoids or enteroids are developed during time. At the day 0, we have our crypt. After 2 days, the crypt closes and takes this spherical shape. At the day 9, the enteroid is fully formed and it starts to develop these little bumps, which are the villi. And the day 15, we can see that the organoid grows in size and it's more complex. For the characterization of the organoids, we use SO1, which is a marker for tight junctions. And also, we use MOC2 to mark the secretory cells. And here we can see that both structures are present in these enteroids, which is indicative that this epithelia is similar to what we can find in the gastrointestinal tissue. Once when we were evaluating the CFTR expression in these enteroids, we see these few cells that had the CFTR expression polarized to the lumen of the enteroid, and these cells are named CFTR high-expressing cells, or CHE cells. So now that we had the enteroids characterized, we start working with the co-cultures. For this, we isolate sensory neurons for newborn pups, and then we mix the enteroids with the neurons and seed it intramatricial and see the interaction of this. This is a schematic representation of what we were expecting. We're expecting that the neuron had direct contact with the enteroid and possibly just innervating specialized cells in the enteroid. Here I show you a video of the neuron marked with peripherin, which is a specific marker for neurons. And we can see the neurites are direct linked to the enteroid, and they have several points of contact with it. Here there's another video where several neurons are surrounding the enteroid, and they have, again, several points of contact with the enteroid. But now we have the question that if the enteroid are attracting the neurons towards it to see if they're secreting something. But here they are mixed in the same dome, so we start using a microfluidic chamber. We are going to denominate it gut on a chip. This kind of systems are microengineered biomimetic systems which replicate key function of units in living organs in vitro. For this, we use this microfluidic chamber, which has 2 separate compartments divided by a little barrier. In this barrier, we have these microgroups, which are 10 micromillimeters in diameter. And with this, we allowed the neurites to cross from one chamber to another, one compartment to another, but not the neuronal body, because they're more bigger, like in 20 to 40 micrometers. Once that we seed the sensory neurons in one compartment and the enteroids in the other, we can see that the neurites cross the microgroups and goes directly to the enteroid. In this zoom, we can see that this enteroid was farther away in the chamber compared with the previous one. And here in the dot lines, we can see that the neurons were directly to the enteroid. Also, we performed immunofluorescence assay, where we can see that once again, we mark the neurites with peripherin, and we can see that the neurites cross the microgroups, and some of them goes directly to the enteroid. Here, delimited by this dotted line. The enteroid is out of focus because it was in a different focal plane, so compared with the neurites. In the zooms, here we can see the neurites crossing the microgroups, and here I show you the distal portion of the neurite that it's directly towards the enteroid. This model, we're going to use it with different CF models like CFTR F508 delete model, and both humanized CFTR wild type and F508. We're going to get this model for Case Western University. We're going to isolate the enteroids, and we're going to seed it along with sensory neurons. Oops. As further steps, we have that we're going to test the CFTR modulators in isolate enteroids and enteroids and sensory neurons interface. We are also going to test the functionality of the sensory neurons in the good ownership system from wild type and CF mice previously mentioned. And we're going to try to load the organoid with bacterial proteins relevant to CF and test neural responses using patch clamping electrophysiology. And in addition to this, the chamber will keep using it for further characterization in this interface in epithelial cells and sensory neurons. As a preliminary data, we're already using different CFTR modulators, and also with this experiment that is for Scoling-induced sweating assay, we can measure the functionality of the CFTR in the organoids. Here we can see that the organoid swells because they're transporting a lot of fluid into it, and here I show you the difference between the vehicle, the DMSO, and the ETI combination. As a conclusion, we have that taken together, we develop a good ownership system to study the gut sensory neuron interface in cystic fibrosis, which could provide valuable insight into the effect of inflammatory signals, microbiome, and CFTR modulators in cystic fibrosis. And also, these systems conserve the architecture of the gut epithelia together with the extrinsic innervation of sensory neurons. And organoids provide a more accurate model for the testing of new drugs, potentially speeding up the development of effective treatments. I want to thank my supervisor, Veronica Campanucci, along with all the lab members for all the help and support for this project, and also the funding agencies. Thank you.
How the neurons exactly finds the organoid?
No, yet. We're going to work on that. Our idea is that the enteroid is secreting a chemioattractant for the neurons to call them, but we're not sure yet. We're going to study it. Thanks. Yes.
Very beautiful model. Thank you. I have actually a technical question. So, in the two microfluidic chambers, is the medium the same?
Yes.
Okay. So the neurotransmitters are being secreted and they go back. And have you tested any? The question is really how the changes. How do you know how frequently you change the media? Is it fluidic and just continues to flow, or it's a static?
No, it's fluidic. It's shared between two chambers, and it's the same media for both. And we change the media every other day, like 2 or 3 days.
Have you tested the effects of, for example, more frequent or less frequent changes to see what the accumulation of the neurotransmitter does if there's?
That's a good idea. In that way, we can see that if they're secreting something to see they're calling to each other. Yeah. Thanks. Yes.
Fascinating system. Excellent talk.
Thank you.
So have you thought about applying, so when the gut on the chip, when that neuron, sensory neuron touches that organoid, I mean, if you could do like mixing experiments with conditional activatable GCAM using like cheek cells, even, like you can mark cheek cells with GCAM and look at how calcium is signaling the different cells of the enteroid change when the neuron, like that's that interface. I mean,
that will be amazing to do. Yes.
Just a comment about the T cells. So we do see T cells are not in the normal mouse. So if you look at the tissue, like published in 2000, that the T cells are after the mouse. So these cells are interacting with the intestinal species with their mouse-by-headed which is a pseudogene in mice, right? So we do see cells, if we use transgenic mice, we do see cells having high levels of CFTR expression, but we have not characterized these cells to be T cells. And I think what you're seeing in your organoid model is what we see in our transgenic mouse models. There are cells that look like T cells, but they're not. We have not confirmed that they are T cells. So be careful with that. Because you're seeing high levels of CFTR in those cells, but you need to really characterize to show that they are in fact T cells. So there is something that happens, I think, in mouse models when you either culture in organoids or you make transgenic mice, then these cells start popping up. But we have not characterized them because in the native intestine, those cells are not present in mouse. In the normal cells.
Okay.
We also see them in the colon in some transgenic animals, but morphologically they don't look like T cells. So there's something that's happening. So we got to be a little bit careful about that.
Okay, thanks. Thanks for your comment.
University of Milano. I'm curiosity. You, in one chamber you put your organoids, major one, and in the other one you put neurons. You use neurons fully differentiated or recursions?
No, they're fully differentiated. Yes?
Culture neurons that are taken from a mouse that expresses CFTR and an android from a mouse that does not express CFTR?
Yeah, we can culture different, from different models of CF and also different models of mouse, yes.
And there's no problem getting them to grow? Or
So far, no. But we'll need to go further into the project to see if there's some issues with that.
All right.
Well, thank you.
Let's thank Oscar and all of the other speakers.